Today we've got Dr. Jason Hawrelak.
Yeah.
Right, yep. From pharmacy. So he lectures into complementary medicine and also clinical practice, I think. So
Yeah.
today he's going to be talking about gut microbiota, which is one of his strong research interests, and letting us know how we can keep it healthy for the gut microbiota and us as well.
Thank you.
Jason.
All right, thanks for giving up your lunch hour time. You could be spending outside in the sun to be here. It's nice. So, yes, I lecture within the Division of Pharmacy, and my PhD was looking at irritable bowel syndrome and specifically the gut microbiota in IBS and the ways we could use prebiotics and probiotics, diet, and even herb medicines to try to alter the gut ecosystem. So that sort of flowed on. Research that started back in 2000, and I had no idea at the time that it was going to become the burgeoning, huge, booming topic that it is today. So first off, I just want you guys to imagine you go to a conference and someone, a research scientist, claims to have discovered a new organ in the human body. And they compare it to the immune system. That's a collective of a number of different cell types all sort of working together, but actually contains 100 times more genes than its host. On top of that, it's heritable, and we can modify it by changing our diet. Different surgery techniques and even different medications can all modify the function of this organ. And whilst you can survive without this organ, when we actually eliminate it, it actually impacts a whole range of different systems functioning as well. And that is a lot like the current situation in which we find ourselves now. And that in the last 10 or 15 years, so when I started back in 2000 doing my honors, we were just making that transition from looking at gut microbiota through a culturing technique of just taking poo samples and putting into petri dishes and seeing what grew, to starting to look at bits of RNA, and that actually and the different genes that are there, and that actually has created the situation that we have now. So it's a few different words for it. Back when I started, everyone called it the microflora or gut ecosystem. Now most researchers call it the microbiota, but some refer to it somewhat erroneously as the microbiome. Something we should know is that it does contain 10 to the 14 viable bugs. And this actually is 10 times the number of cells that there are in the human body. And in a typical Westerner, you're looking at about 1,000 different species. And back when we were only looking at culturing techniques, we were looking at maybe 400 or 500 was the estimate at the time. But there are non-Western populations that have a lot more bugs than this. This typical healthy Westerner, about 1,000 different species. And generally you could say that when we're not stressed, and we're eating well, and we're not taking a range of medications, which is probably not that common, we actually live in a mutually beneficial symbiotic relationship. In which case, that organ is actually promoting our health. But when we do other things like we get stressed, we eat really poor sort of diet, or certain medications can actually alter the functioning of that ecosystem and then therefore be involved with disease, a bit like our immune system where it can go a bit out of whack. And that's what we would call dysbiosis. And that was a term that was probably not so widely used in early 2000s, but it's very widely used now. And I found it amazing. I went to a gastroenterology conference in Rome 2 months ago, and just to hear these like high up gastroenterologists talking about dysbiosis and a few other concepts that 10 or 15 years ago they wouldn't have, because of the way that technology has advanced and research into that area has advanced. Is quite amazing. So to me, the idea of the gut microbiota being a vital human organ was put forth initially by a Swedish researcher called Stig Bengmark back in 2001, around that time. And it didn't necessarily take hold that well, but it's slowly sort of going through. But it sort of resonated well with me. It's like, oh, that's an interesting way of looking at it. And it's certainly well and truly taking hold now. And we can look, if you scooped all this bacteria out, if you could, and put it onto a plate, you'd get basically a kilo or 1.5 kilos of gut bacteria. But we now, though, in terms of its genetic potential, that actually rivals the liver in terms of biochemical reactions in which it can actually participate in. So it can very much be seen like an organ. Now, what's been put forth in literature more recently is more of an idea of humans as a superorganism or meta-organism, of which you are 10% non-microbe and 90% microbe. And I actually think that the more research we're actually doing, the more we'll actually be moving towards that model. And if you think that, it actually changes a lot of ideas and a lot of concepts if we actually start looking at our body as being 90% microbes and 10% non-microbes, for us being 90% microbes. But that's just seeping out there. So to give people an idea of this gut ecosystem, generally starts in the oral cavity. We can see it's actually quite heavily populated, 10 to the 8, 10 to the 9 cells per mil. The stomach is relatively empty. I mean, probably the only native inhabitant would be Helicobacter pylori. And the rest are what they call allochthonous, which you can see as travelers. And most of the travelers have basically just come from saliva that's been swallowed. But they're just generally there in relatively low amounts. So you can see here in the stomach, you're looking at 100 to 1,000 bugs per mil of content. And as you move down into the small bowel, it becomes more and more dense. Then as we get to the colon, you can see it's where it's immensely dense. And this is where the population diversity is actually far greater in the small intestine, where you've got a good number of indigenous microbes as well as travelers. But in the large bowel, it is very much dominated by the indigenous bugs that are already there. And really what researchers consider to be the most important component of that whole gut microbiota from mouth to anus is the colonic microbiota. And I think there's a couple reasons for this. One is obviously just the density. There's a lot more bugs there per mil of content, hugely more than anywhere else in the gut. Secondly, transit time has slowed down a lot. Yeah, the transit to the stomach and small bowels relatively quickly. And these microbes tend to be eating the food that we've eaten, or the foods in the colon that we don't digest so much for the most part. And when that has a chance to slow down a lot more, because in the stomach the food might be there for 30 minutes, 60 minutes, in the small bowel can be up to 3 hours before it sort of passes through the colon. But then from there, in someone who's what I call healthy, gut transit time might be 16 hours or something in there. Someone who's not quite so healthy might be 3 or 4 days or more in that colon part. So there's a lot of chance for those microbial byproducts that from when they're consuming foods and their byproducts that come out to actually interact with us. And researchers have divided this into a sort of semi-artificial but potentially harmful versus beneficial groups. And that's what I've got here. And obviously this is a gross simplification because you're talking 1,000 different species, and here there's 17. So a big sort of simplification. But it sort of gives you an idea of what bugs are less common at the top versus most common down the bottom. The bugs in blue are generally, in most circumstances, considered to be beneficial. It doesn't mean that if you injected them into the brain or something, that wouldn't cause harm or an infection, but generally when they're in the gut, they're actually not being problematic. The bugs on the left would generally considered to be more potentially problematic, and the bugs right across the center here produce beneficial compounds that improve our state of health, but also can produce compounds that aren't so helpful for us too, depending essentially on what we feed them, so what the balance is, and what the environment is like, which is mostly pH related. And if we would have looked at this back when I started, back in essentially 2000, most people were just talking about 2 different species as being the primary beneficial ones. Lactobacilli, which in common parlance, in the general public, they just call acidophilus, and bifidobacterium species, which they would just call bifidus, would be the 2 that people were focusing on more so. But since we started using more sensitive molecular techniques, we now know that there's a few other bugs that actually are key players for a healthy gut ecosystem. The Faecalibacterium prosnitzii, Roseburia, Eubacteria, and Rubinococca are actually there in very large numbers. In fact, Faecalibacterium in a healthy gut actually makes up 15% of what's there. But before we were using molecular techniques, we didn't even know it was there. So we sort of defined a little bit, in a few slides, what the microbiota is, and so the beneficial members versus those that aren't. But I think it's important that we actually know what this organ actually does do for us. What sort of roles does it play in your health? Well, we know it's very key for actually modulating the immune response. And this goes back to research done, I think, in the 1960s and 70s with rat models, where they could actually sterilize the GIT and then keep the little rats in sterile cages so they were not able to re-inoculate themselves with any bacteria. And then look what effect that would actually have on their systems. What they found in rats is that their thymus gland shrinks. Their spleen actually shrinks, and their ability of their white blood cells to actually deal with invading pathogens goes down markedly. And the number of IgA antibody-producing cells in the gut goes down by about 90%. You reintroduce the bacteria back in, spleen grows, thymus gland grows, white blood cells start functioning properly again. And researchers are now talking about the gut microbiota is actually important for training our immune response to behave properly. in this age of allergies where we're overreacting to things like peanuts and pollens, they're certainly thinking that this dysbiosis, that is meaning that the immune system doesn't get trained to react properly to potential allergens. What we call normal gut motility is also very much related to these bacterial visitors we have in our gut, or inhabitants in our guts as well, and that the same sort of animal models where you eradicate the bacteria, the entire gut transit time slows down markedly. So it goes much slower from mouth to anus. Phytochemical metabolism. So phytochemicals are those sort of healthful compounds we find in fruits and vegetables, whole grains, and legumes, and those bright colors. And blueberries is sort of the blue antioxidant compounds. Well, it turns out that for us to actually get benefit from polyphenols, phytoestrogens, and other sort of plant glycosides, it actually depends on the gut microbiota to actually alter them first. Otherwise, they just pass straight through you unabsorbed in a way that's not actually remotely helpful. So bugs actually will essentially eat the sugar component and release the medicinally active aglycone, essentially, which we then absorb. They also produce things like short-chain fatty acids and polyamines, both of which I won't go into great detail, but they actually, you can see them as almost fuel for your intestinal and colonic cells. So in terms of promoting intact epithelial tissue, they're very important. We'll look at some in greater depth. The microbiota plays a huge role in our overall digestive processes and nutrient absorption. In fact, you might be surprised to know that 10% of your daily energy needs are met from basically the microbiota fermenting fiber in your gut. And in our closest sort of living relatives, the sort of lowland gorillas, they make about 30% of the daily energy needs through that, with a gut that's pretty much identical to ours. And in cultures where they eat probably plant-based, high-fiber diet, it goes up to around the 20% mark too. So we certainly have a great propensity to develop that ecosystem if we feed it the right foods. And essentially this is mostly from making short-chain fatty acids from unabsorbed carbohydrates. And carbohydrates in this instance means the broader category, not just like sugar, but because fiber is carbohydrates. It's just have a certain bond that we can't break down, but bacteria do. And that also includes all these oligosaccharides and resistant starches and pectins and mucilages. And they also enable us to absorb more calcium and magnesium and trace minerals too. And they do this essentially by changing the pH. And when they change the pH, it makes those minerals much more soluble in the colon. And other of our microbial inhabitants produce vitamins, like B vitamins and vitamin K as well. And that's why there are case history or case reports published of people that have been on long-term broad-acting antibiotics who get bleeding disorders, because the vitamin K production has been impacted enough. And B group vitamins too. And interestingly, research in the last couple of years has actually found pathways for some B vitamins in colonocytes that we didn't know existed before too. So we've actually evolved to be able to get some of our B vitamins from microbial production. Unfortunately, not vitamin B12, which would be good if we had developed that, but we haven't. But other B vitamins we do absorb actively. Colonization resistance, one of the most important ones. So, as you know, every single food item that you eat, or most things that you drink, will contain potential pathogens. Yeah? And some of them will be obviously destroyed in your stomach when your stomach acid is good. But a lot of it, of our resistance to those infected pathogens, comes down to having a healthy, intact microbiota. And they've done research in animal models here too, where they can give a rat a dose of antibiotics, and they compare how many bugs of salmonella, for example, will cause diarrhea with the antibiotics or without the antibiotics. And sometimes you can give 10 times less of an infecting pathogen after an antibiotic to actually get an infection. It does this for a few different ways. One, they release antimicrobial substances. Two, they compete for adhesion sites in food. And I like to tell my patients is thinking they've got a bit like a parking lot. And when your ecosystem is healthy and robust, every single car spot is taken. And that means that any ingested pathogen, well, they're just going to go right through because there's no place to park. It's like I had a hard time finding a parking spot today, so you just drive around and around. But in this case, they have to leave the anus, so they're gone. And the metabolic end products, again, those short-chain fatty acids alter the pH. And in general, a lot of pathogens don't like a more acidic pH. So by keeping the environment in the colon more acidic, it means that there's not a chance for the pathogens to actually thrive. And more interestingly, weight management. And I think this is one that was certainly surprised me when the research first came out. And this was mostly done by Turnbaugh's experiments in 2006. But we now know that the microbiota plays a vital role in energy homeostasis. And there's in obese individuals, their microbiota has a greater capacity to actually extract nutrients out of their leftovers. And perhaps even more interestingly is that the obese microbiome is transmissible. In fact, they can give fecal matter from an obese rat into a non-obese rat, and that rat will become obese. Despite the fact they're not changing their caloric input or output. They can make this one turn into that just by changing the microbiota. And this was Turnbaugh's experiment, which really threw everything on its head because we didn't ever imagine that the microbiota could have such an impact. And there's now been case reports too of people that have had fecal microbial transplant, probably for Clostridium difficile infection, that have ended up becoming obese. And it was the donor, it turns out, was obese. So people are starting to look at donor characteristics more carefully now than what they once did. And mood. And this is an area, again, that we wouldn't have necessarily thought too much about 10 years ago. But the microbiota modifies mood. Now we know that, again, those germ-free rat and mice models tell us that these guys have got different brain chemistry compared to conventional mice. But when we actually put bacteria back in, their brain chemistry and behavior normalizes. And there's also been research taking fecal matter from anxious humans and putting it into rats, and those rats become anxious, solely because of the change of the gut microbiota. How is that done? No one's totally sure yet, but it might be because different microbes can produce GABA, or serotonin precursors. They can change the levels of inflammation. They can actually stimulate the serotonin release, and they can interact with the vagus nerve. All possibilities of how they can actually interact with mood. And there has been some recent research using probiotics in anxiety, showing that they were effective, preliminary research, and even using prebiotics, which we'll discuss later on, things that just change the ecosystem and people's anxiety scores coming down. So, hopefully that's given you a very brief summary of the important role that the microbiota organ actually plays in our health. And you could do a whole 2 hours lecture just on this. Or in fact, there are whole conferences on this. It's a very brief introduction. So now that we know that, and why it's important, and why we want to make sure it stays as healthy and well-nourished as possible, we should look on to things that actually cause its harm. So what agents cause harm? Well, not surprisingly to most of you, antibiotics would be top of that list, but chemotherapy does too. And I think it was believed in the past that chemotherapy, often you get this long-standing diarrhea afterwards and during, it was caused to sort of collateral damage to enterocytes, gut cells, but it turns out that it does that too, but it actually causes a big shift in the ecosystem of the gut too. Pretty on par to what we get with antibiotics. Stress, big factor. And that we know when people go through highly stressful periods that the beneficial bacterial count in their guts tend to go down. Animal models and some human studies too. And potential pathogens, or what they call pathobionts, tend to go up at that point too. Radiotherapy, similar to chemo. But the last two are actually fairly recent research, 2014. So this research has not sort of disseminated through to clinicians as yet, but it turns out that proton pump inhibitors cause a massive reduction in gut bacteria diversity. Massive. In fact, people that have been on PPIs for a great period of time, it was pretty similar to what you had with long-term multiple courses of antibiotics.
What are PPIs?
So proton pump inhibitors.
Yeah. So why would you be on them? I know NSAIDs are non-steroidal.
Okay, so yeah. PPIs are used for reflux,
Okay.
mostly, and for gastritis and Helicobacter pylori infection, but mostly for reflux. And they're one of the most common class of medications used in Western nations. Millions of prescriptions every year. And now they're actually sold over the counter too in Australia. So they're very widely used. And this research is relatively new, but it's showing that pretty massive diversity changes, even from a week of taking it, and even in short-term use, a month afterwards, it's not quite up to what it was before. And it turns out that proton pumps, for us, actually pump out stomach acid. So when we sort of inhibit them, that stops that. But it turns out bacteria got proton pumps too. And when we inhibit theirs, they die. And then eventually it just means that those that can't actually adapt to that die out. And then this recent research showed non-steroidals cause sort of the beneficial blood count goes down and the pathobiont count goes up. In fact, and they're actually linking in the damage to the gut that's actually caused by NSAIDs very much to the dysbiosis that's caused by NSAIDs in research from this year. And there are also some sort of agents that alter the whole development of the gut ecosystem in negative ways. First one is C-section birth. And we know that kids that are born by C-sections have completely different gut bacteria for their entire lives, essentially, to those born vaginally. Yeah, they're more likely to get a whole autoimmune disease and celiac disease, a whole range of things. And it's because they missed that transiting through the birth canal. And we know now that the vaginal ecosystem actually changes about 3 weeks before the birth process. It actually changes dramatically to have higher counts of beneficial bugs. So but the baby would usually go through there, and the head's coming through, actually, usually some feces comes out too. So it's a perfect inoculation system, essentially, that we've sort of bypassed. And what the bugs who are born by C-section do, they get hospital staph bugs. Hospital bacteria like C. diff is far more common, Clostridium difficile in C-section born kids, and skin bacteria. And that seems to have a whole bunch of flow-on effects. That initial colonization seems immensely important. And following that is formula feeding 2 actually induces changes to the whole sort of gut ecosystem, because breast milk is not just nutrition. There's 1 billion bacteria per liter of breast milk. And that are actually, the gut actually samples different bacteria and we now know transfers it up to the breast milk, which is pretty amazing. And there's actually different sugars in the breast milk which selectively feed certain bugs to actually modify the environment in a way that you can't do with formula. So it actually alters their flora.
So is there any way to sort of, you know, if you've had a C-section, you've had a C-section, is there any sort of protective clutching at straws?
No.
No.
But there is some research, and I think the first study is about to be published, done in Puerto Rico, where they've actually done vaginal swabs for kids born C-section. So the first thing the kids get is vaginal mucus smeared around here into the oral cavity, and their flora seems to develop normally.
Wow.
So there is ways of getting around it. It's just that it hasn't filtered down to the clinician aspect yet. All right, so we covered those things. So we'll move on to dietary factors that can negatively impact the microbiota. One is preservatives called sulfates and sulfites, which are used in sort of non-organic dried fruits. A lot of red wines will have sulfites in them as well, and a lot of just junk food in general does, including those those prawns you can buy at the supermarket you can get from Bangladesh, that come from a long way, that they look fresh, but they're not fresh. It's because they're covered with sulfites. And it turns out these sulfites very selectively feed up a group of bacteria called sulfate-reducing bacteria, which then produce a toxic gas called hydrogen sulfide gas, which causes a bit of damage to the intestinal tract. And it's the amount of that byproduct produced is very much related to how much sulfur-based preservatives you consume. High protein diets actually can cause massive negative shifts in the ecosystem. And particularly ones that are those sort of high protein, low carb diets, which essentially cut out a lot of fiber is the byproduct. So you end up starving a lot of the beneficial bacteria. And you can get a 50% decrease in beneficial bifidobacteria counts just in 4 weeks in such a diet, as well as some of the other bugs that I mentioned before, like Faecalibacterium, Prospecia, etc. They go down a lot in that sort of diet. And high fat diets too. That people used to think that, okay, well, bacteria aren't utilizing the fat. And in general, fat aren't that well utilized, but when we have a higher fat intake, we secrete a lot more bile to deal with the fat, and it turns out there are certain bacteria that like eating our bile. So we increase their numbers, and those bugs tend not to be so good. Bilophila wadsworthii, Bacteroides that produce more sort of potentially toxic byproducts, go up when we go on a high fat diet, and beneficial bugs tend to go down, essentially because we're starving them. Dietary emulsifiers, which are widespread in processed foods. Ice cream's got a lot of them, not necessarily the natural ice creams, but the ones that you would tend to buy at supermarkets. That actually causes, this very interesting study by Chesing in 2015 was looking at dietary emulsifier, even at low amounts that you'd find in dietary amounts in rats actually thins the gut mucus and caused increased risk of obesity and metabolic syndrome, type 2 diabetes, and cause a bit of inflammation in the gut too, and it changes the gut ecosystem as well, and that does that by thinning the mucus, the protective mucus layer, because some bugs eat that mucus, as it turns out. Some good bugs do. Sucralose, an artificial sweetener, tends to decrease levels of beneficial bugs. And essentially diets that are high in refined carbohydrates, and I'm stressing the refined component there, because what are refined carbohydrates, but essentially we're able to extract everything out of it. There's nothing left over to reach your colon to feed the bacteria. So essentially, it's low in what I would call microbiota foods, or other researchers call it microbiota accessible carbohydrates, or MACs. And as Sonnenberg states, we're essentially starving our microbial self when we're eating diets like that. Because we're not nourishing that 90% of us that actually resides in your colon when you're eating a really refined diet. So how can we nurture and protect the gut microbiota? Obviously, we can avoid those factors that I've highlighted to the best of our ability. We can't do anything if we were C-section and formula fed. We can't change that, but we can try to work with where we're at now. And we can encourage consumption of foods that actually nourish and support the growth of the good bugs in the microbiota. So how we can avoid those agents. So obviously we can work on minimizing medications. There's lots of reasons to avoid overuse of antibiotics. They save lives, they save limbs. There's times when their use is absolutely needed. But we know from the data that they're overprescribed. They're even still being commonly prescribed for viral infections in clinical practice. Assess alternatives to NSAIDs and PPIs. Because sometimes it's a matter of people changing their dietary exposures. Like you can often modify people's heartburn symptoms by making a few small changes to diet and lifestyle, which I would think would be a good thing to do first before using PPIs. And we also need, and again, chemotherapy and radiotherapy are essential treatments when they're used. So what we have to do is try to minimize the damage when they are being used and help restore the damage afterwards. We can manage stress. We can try. Exercise, daily meditation, breathing exercises are all things you can do that will actually help modify that stress response. And minimize those negative dietary factors. So avoid any sort of processed refined foods. That means that you essentially will get reduced intake of dietary emulsifiers, reduced intake of refined carbohydrates. If you don't eat that, you'll tend to have a much happier gut. Avoid high protein or high fat meals without dietary fiber, and I think the dietary fiber is the key thing. It's not like, I know some people do go better on higher protein intake, which is, I think everyone's a bit different. Some people are fine on low, some people go better on high, but you still have to have lots of fiber with it. And there are people who are taking a ketogenic diet to help manage their epilepsy, for example, and there's some good research there suggesting it's a rational approach to actually take, which means that they're just having huge amounts of fat with very little else, and that does cause major shifts in the ecosystem that would not be considered beneficial. But in that case, you'd supplement fiber or something to try and offset that. So there's ways of looking at it. But the key aspect seems to be dietary fiber and preventing the problems that you often get with high fat and high protein. So I suggest you aim for every meal to consist of mostly high fiber plant foods. And we know that sort of offsets most of the potential problems with such a dietary program. So that, make it more like that. So you're still getting, brown rice, lots of vegetables, sweet potatoes, carrots, a whole range of different sort of food for your microbiota carbohydrates in that sort of meal that you don't get with that. But there are some specific foods we can use that sort of are perhaps more nourishing for the microbiota, which I'll cover shortly. But I think the important bit is having microbiota feeding foods daily and preferably with every meal. Whole plant foods, polyphenol-rich foods, resistant starch, prebiotic-like foods, and FOS and GOS. And I'll go through each of these because some of you know what they are, some of you won't at this point in time. But essentially, you want to eat mostly plant foods if you want a healthy gut ecosystem. And sometimes you have to tweak that around when people have got very sensitive guts to gas production, for example. That's pretty common these days. You have to be gentle with how you introduce it, and there's some fibers work better than others. There's ways of tweaking it, but essentially, we're still trying to aim for the same thing. So whole food, minimally processed, probably plant-based, moderate fat, low in saturated fat, specifically moderate protein, rich in variety. And I try to get my patients to consume 40 plus different whole plant foods a week, not a day, a week. So buckwheat would be one, brown rice, red rice, black rice. So 40 gives them a target, because if otherwise they just have more variety of plant foods per week. Doesn't necessarily mean much, but they have a target to actually aim for. It's better. There's no research on the 40 plus. That's just what I think is doable. That seems to create a wider diversity of plant foods. Because research shows clearly that the wider diversity of plant foods that you eat, the more diverse your gut ecosystem is. And the assumption here is that all the ecosystems in nature that are healthy and diverse we associate with, or that are diverse we associate with health. And I think it's the same idea with the gut ecosystem, that diseases tend to be associated with low diversity. And when people, and health seems to be associated with greater diversity. So to increase diversity, we need diversity of plant foods. And essentially because you're getting lots of different sorts of fiber. And fiber is just not one thing. There's lots of different shapes and tweaks to different molecules, which means that you feed different bugs by having wide diversity. So, so base your diet, whole grains, vegetables, fruits, legumes, nuts, and seeds as the core of the diet, and other things like fish and chicken and meat around that bit, but those things being the core. Eat polyphenol-rich foods, because as a general rule, foods that are high in polyphenols beneficially shift the gut ecosystem. So not only do you get the antioxidant components in those polyphenols, it tends to shift the ecosystem in a beneficial way. So black elderberries are top of the list, but they do grow well down here. You just have to be able to wildcraft them because they're not commercially sold. But black currants, mine are just ripening up now, so that's nearly the right season for black currants. Very rich in polyphenols, more so than blueberries in there. Cherries, strawberries, we're coming to the right season, besides apples, but other than that, we're coming to the right season. Nuts and seeds like flaxseed meal, chestnuts, and it's essentially those sort of dark compounds, or the purple compounds or red compounds, they're the polyphenols. Choosing purple carrots instead of orange, purple potatoes, or red potatoes instead of white. Broccoli, red onions, spinach, red cabbage, those sort of purple and reds are very important. Black rice, red rice, these days I eat a lot more of these things than I ever used to before. Black olives and olive oil all contain those polyphenols. And I'm going to talk about some what's called prebiotic-rich foods first, but I should define prebiotic first. Are any of you familiar with the term prebiotic? We all know probiotic, then it's probably quite familiar. Prebiotic is quite different, and that it's a very specific definition, but it's a non-digestible food ingredient that, when consumed, essentially changes the ecosystem in a beneficial way, in a way that benefits your health. Would be a way of looking at it. But it is selectively fermented, so you can see it almost like being a selective fertilizer. If you've got weeds growing in the garden bed and some vegetables, imagine if you could just throw some fertilizer that would make your veggies grow, but the weeds couldn't use. A prebiotic is very much like that. Only certain bacterial species can actually eat prebiotics. So those of you who are old enough, like me, who remember Pac-Man, and I found that the other day, and I thought that's an interesting. It's a little bit wrong, but it's pretty close. And that you can see these Pac-Man as being the sort of normal, healthy, beneficial bugs in your gut. You're giving them selective fertilizer, and then they can deal with the bad bacteria. So there are certain foods that are rich in prebiotic compounds. And there's two main categories. One is fructooligosaccharides, which people call FOS for short. And the other is galacto-oligosaccharides, which GOS for short. And FOS, very high in garlic and onions, really, really high in Jerusalem fartichokes. So high, in fact, that some of my colleagues call them Jerusalem fartichokes, because you do get a lot more gas production. But they're about 20% inland, so very high. And legumes in general, beetroot, pumpkin seeds, sunflower seeds, have got small amounts of GOS as well. And there will be certain people that don't tolerate this category of food well. Those who are on the low FODMAP diet do not go well with those foods because they are fermented. And when they ferment it, they produce gas. And when people with IBS in general tend to not tolerate intestinal gas production very well. Yeah. But for those of us who do, they're very healthy foods that actually increase the growth of beneficial bacteria in your gut. So I tolerate them well, so I will eat these foods daily. But my patients with IBS, not initially anyway, will not eat those foods. And there's also what I call prebiotic-like foods. These are foods that in human clinical trials actually increase the growth of your beneficial bugs in the gut. But they tend to be more, perhaps widely or better tolerated than that other category that contain FOS and GOS. Brown rice, black currants, carrots, dark cocoa, almonds and green tea, all in human studies increase levels of beneficial bugs and often decrease levels of pathobionts in the gut. And in fact, with all these foods, it's actually the almond skin because it's high in polyphenols. Cocoa because it's high in polyphenols. Green tea because it's high in polyphenols. It's a polyphenols element of those foods that tends to be beneficial. So eating skinned almonds doesn't work. And the last sort of food category we'll look at in greater depth is resistant starch. And most of you will know starch, yes. Resistant starch just means that that starch molecule is resistant to your digestion. Luckily, it's not to your microbes. Your microbes will very happily eat it. but you won't get any direct nutrition out of it. They'll produce you with nutrition for eating it themselves. So legumes, bananas, particularly green ones, the greener they are, the more resistant starch they have, but they're also a lot less palatable, so you have to find a little, somewhere in the middle where it's okay. Cooked and cooled potatoes, particularly roasted, once they're actually cool the next day, are very high in resistant starch. But when you eat them fresh, they're not so high in resistant starch. So I tend to cook mine and eat them the next day, and that way I know my gut bacteria are being fed from that consumption. Other root vegetables in general, like cassava and sweet potato, rye bread, oats, particularly uncooked oats. Porridge is not a very good source of resistant starch, because when you're cooking the oats, they sort of everywhere, and their sort of starch goes out there, and you can easily digest and break it down. But when you just soak the oats overnight, or even just for an hour before eating it, the starch stays in there in that resistant form, and it means that you're feeding your gut bacteria. And it's actually much better from a glycemic load perspective anyway, that the soaked oats will give you energy for far longer than cooked oats will. And cashew nuts too. And that's just a small list of foods that you can think of adding or tweaking to your diet. And there are specific supplements too, which are prebiotic supplements, which I won't go into great detail, just to say that they are actually out there. Lactulose, which is commonly used as a laxative in large doses, is a prebiotic in low doses. Fructose oligosaccharides are the same that's found in the dietary sources. Then we also have GOS, which we find you can get in a supplement form, which is a slightly different configuration of the molecule than what you find in food forms. In fact, supplemental GOS tends to decrease gas production and decrease bloating, whereas food sources of GOS increase gas production, increase bloating. Yeah. And last but not least, partially hydrolyzed guar gum. But guar gum increases gas production, hydrolyzed guar gum decreases gas production. So sometimes it's a matter of just small tweaks to the molecule means it feeds different bugs. So we'll go back to that diagram, and we can just add in different things. So prebiotics feed lactobacilli and bifidobacteria, and feed Calibacterium parasitii as well. Prebiotic-like foods, bifidobacteria, lactobacilli, polyphenols, same two bugs. But then resistant starch, we see feeds all these bugs down there by the bottom. So the idea is, it's not that you just eat one of those food categories, but that you eat something from all those food categories on sort of a weekly basis. And obviously you can't necessarily achieve that on a daily basis, but the idea is you're just trying to have that wide diversity of foods on a weekly basis. Yeah, if you can do that, your gut microbiota will be happy. So that is a good microbiota nourishing meal. You've got black and red rice, you've got purple potato, you've got sweet potato, you've got some shallots, you've got some beans. So essentially you're covering all the, you've got polyphenols, you've got resistant starch, you've got galactose oligosaccharides, fructose oligosaccharides, all in that one meal. So there's certainly a huge trend at the moment going with fermented foods. And I should put a caveat here that I'm half Ukrainian, half German. So my parents grew up with huge vats of sauerkraut in the basement. I love sauerkraut, and I've been making it for 15 years. But there's certain things it doesn't do, which online you wouldn't, if you read the blogosphere, you'd get a different perspective on this. But listen, they don't feed that microbiota organ. I think people sometimes get this idea that when you eat sauerkraut or something, your lactobacilli, your own good bugs will grow. And it's like they're not cannibals. They're not going to go, oh, here's another lactobacilli, I'm going to eat that and become super strong. But there is this idea that foods, sources of probiotics, or supplement sources will permanently repopulate or grow in the gut. It turns out that's not right. We had, in fact, 30 years of research telling us that any sort of exogenously supplied probiotic agent from those two genera, food or supplement, does not permanently stick in your gut. If you can stay there for a couple weeks, you're doing very well. It means the strain has been well chosen. Some of them will pass straight through you altogether. So we know that we cannot re-inoculate, we cannot repopulate, we cannot populate. I'm trying to stress that point, because I think that misinformation is out there, and it means that people neglect the factors that we know do encourage a healthy gut for things that aren't so important. So any colonization you get is only temporary. Why is that? Because your gut flora is as unique as your fingerprint. Completely different from anyone else in this entire room. Yours will be similar to your brothers, your sisters, very similar to your mum. If you're born vaginally, very, very similar. But then it's been modified by your dietary factors, drug use, like antibiotics, etc., will have actually changed where it's at now. which is why even like identical twins that essentially would have been colonized, essentially very similar initially, but can actually have different microbiota. It's not as different, they're actually more similar than anyone else in the entire world, but they're still different. But what it means is that when these bugs that we take in supplements or food forms, they can't seem to stick. And we don't necessarily know why that's the case. Because we know that fecal transplants can help in certain situations, and some of those species stick, but these two don't seem to have the capacity to stick. And before we finish off, we'll just go through an example, because I know all of us take antibiotics at different points of time, and I think this is the intervention that we know has got the sort of greatest negative impact on the microbiota. So what do we do when we actually have to take it? We want to make sure that we do have to take it, but the other thing to know is that every antibiotic is different in terms of what its breadth of action is. And because your gut ecosystem is different, it means that the impact it will have on you will be different than it will have on someone else, potentially. We can look at research that had 25 or 30 people. Most of this research is done in small numbers because there's not much profit to be made from researching the impacts of antibiotics on the gut microbiota. So the studies tend to be small. But we can look at what happens to the majority of people and try to get an idea, extrapolate that across to you or to your patient. And I did a systematic review when I was doing my PhD, looking at all the data on all the antibiotics' effect on the microbiota back in, I think it was 2004 or 2005. And most of that data, at that point, still relied on culturing techniques. When you look at the data, it looked like the microbiota was different from maybe 7 days up to 6 weeks, 42 days, for the worst antibiotic. And then it seemed to look like it was pretty normal to what it was before. And at that time I thought, oh, listen, maybe antibiotics actually don't cause such a significant shift. If only for a week or 2 weeks or 3 weeks, it's not really a big deal if it goes back to the way it was before. And that, certainly at that time, was what evidence was suggesting. But I did come across one study that suggested that after a single course of antibiotics, the microbiota functioning was different for up to 16 months. So that's the ability to break off those polyphenols and those phytoestrogens so you get those sort of healthy compounds. That differed for 16 months after a course of antibiotics. You're like, so it looked similar, but it actually doesn't function normally again. But when they started using much more sensitive molecular techniques, looking at 16S rRNA, it actually told us a whole different story. One, we could see that antibiotic-resistant bugs were there 4 years after a course of antibiotics. They're still there. The resistance genes are still there. But also, these alterations actually don't just last for 1 week or 6 weeks. Clindamycin, which is probably the single antibiotic that causes the biggest shift, 1.5 to 2 years, the gut ecosystem still does not look the same. Triple therapy, which they give for Helicobacter pylori infection, 4 years, the ecosystem still does not look the same. Now the core, what people would call the core ecosystem, tends to return. It's the peripheral bugs that don't. So essentially what you have is localized extinction. So in that person's gut, whole species are gone completely. And the part of the issue is here is that some of these species, we don't know what they do. We don't even know that they exist. So we didn't know where they exist until recently. There's one bug called Oxalobacter formigenes that these days is about 30% of Westerners that actually have that bug in their gut. When they take Helicobacter pylori triple therapy, 2/3 of people, it becomes extinct in their gut. Now it turns out that bug plays a pretty vital role of eating dietary oxalate. So it lives in your colon, it eats oxalic acid that we find in a whole range of different vegetable sources. And when it eats that oxalic acid, it means that we don't absorb it. Because when we do absorb it, it actually causes kidney stones. So when that species is gone from people's guts, that person has a much greater risk of having kidney stones for the rest of their lives, essentially. But what's really interesting is, let's say we treated a woman who is 22 for H. pylori, we got rid of it, we got rid of Oxalobacter formigenes too. Her kids will not have that bug in their guts. So that whole family line, from that point onwards, that bug will be absent, and that whole family line will have increased risk of kidney stones from that one sort of action. So there are repercussions that we're just starting to tease out now that we can actually see things more clearly. So essentially, our aim when we're taking antibiotics is to minimize and restore the damage. So again, we want to minimize antibiotic use. We want to support the microbiota during and after use. So think of the gut ecosystem, I like to do it as a tropical rainforest environment. Every single metabolic niche is used. Very, very diverse. Life everywhere. Boom. And that would be something like clindamycin or triple therapy where you're taking a couple of antibiotics at the same time. It causes massive shifts where you can often have a millionfold decrease. So for every million you had, there's one left. So massive shift. Not all antibiotics would do that, I should point out. But with bugs, it more look like that to that. So essentially we want to protect, there's two things we want to do. One is protect the microbiota during antibiotic use. So how do we do that? We know that the right probiotic actually reduces antibiotic-associated side effects. That's good. It actually attenuates the damage to the microbiota. That's good. And it prevents the growth of antibiotic-resistant bugs. So you've got a triple whammy, that people who take the right probiotic during antibiotics get less side effects, less damage to their gut, and less antibiotic-resistant bugs. On a societal level, that's huge. But strangely enough, it's still not common practice enough to actually do this, even though we've got meta-analysis, systematic reviews showing that it's not low-level data, it's just high-level data, but it hasn't filtered through to clinical practice. But that said, you can tell your friends, and if you have to take antibiotics, know that it actually makes a difference, and the data is there. So you want to make sure you take it during antibiotics. You don't get those benefits if you wait till afterwards. You get the benefits if you do it during antibiotics, and I suggest my patients do it for 6 weeks afterwards, because the definition of antibiotic-associated diarrhea is any diarrhea that happens for 6 weeks after you stop and take antibiotics. So that's that window where there's the biggest amount of change. And the simple thing you can do, and this was a study done here in UTAS, put up in Launceston, and they actually had people eat Valia yogurt. And it has to be Valia yogurt, because this particular brand of yogurt's got 3 different bacteria in therapeutic amounts. And they found it reduced the risk of antibiotic-associated side effects by 78%, just by eating a serve of Valia yogurt daily. Massive shift. And I usually, when I'm taking antibiotics, I will eat it 2 or 3 times a day, but then afterwards at least once a day. And as long as you got that half cup, that's the minimum therapeutic dose, because you actually get 100 times better survival in a dairy medium than you do with any other sort of medium. So you can get a lot less bacteria. So half cup of yogurt is equivalent to taking a capsule of something else, if it's the right yogurt. So a very simple thing to do. Our second job is to help the survivors post-antibiotics. But probably not those survivors. We might leave those ones. We want to help these little guys, these little isolated bifidobacteria that have survived the antibiotic onslaught, grow from that to that. So we need to nourish and support this and feed them. So we give those same foods that I talked about before. And I think this is very much a clear need for prebiotic supplementation at this point too. So you take that for 6 weeks post-antibiotics, and any one of those 4 will help be that sort of selective fertilizer that will encourage their regrowth more quickly. Because bacteria, unlike us, don't need to have sexual intercourse to reproduce, and they reproduce really quickly if we give them the right food and the right environment. So you can have just those 1 or 2, if you have 1,000,000 to 1 sort of drop in bug count, you still got a couple thousand of those good bugs still there. You just have to help them regrow into their normal healthy levels, and you can do that if you give them the right food. That changes the pH, so you give them the right environment for them to thrive in. And I think my last slide is really just this reminder of trying to make this shift in people's thinking that you're really a custodian of the microbiota, because it's quite a fragile organ, and it gets passed on in a bit of a unique way. It's really that whatever the mom's gut flora is like at that point when the baby's born has a huge impact on the health of that infant the rest of its life. And what we've seen in Westerners is a massive loss in diversity. The Hadza is a hunter-gatherer tribe in Africa who probably live a lifestyle much more akin to what our ancestors did. And they eat, on average, I think 100 grams of fiber a day. Typical Westerner, 15 grams a day. A lot more fiber. Now, when we look at and they don't take antibiotics or PPIs or NSAIDs. They're all born vaginally, and they're all given breast milk. Their ecosystem, they've got 3,000 different species in their gut. We've got 1,000. And they recently isolated some fecal specimens from a tribe up in Upper Amazon that had no contact with Westerners, 4,000 species. So we have essentially lost, people are arguing, between 2/3 and 3/4 of our diversity in Westerners. Why is this? Well, 3 generations of antibiotics, widespread use of PPIs, NSAIDs, even PPIs during pregnancy, because that initial colonization, as I said before, is very important. Western diet, C-section birth, antibacterial everything. has all impacted that diversity. And as I said before, we're just starting to tease out what the implications of the loss of diversity are. Because using new tools, we can actually see what's there, or what used to be there, if we're looking at these other sort of samples. And it's great that there's researchers going out there and sampling those other pre-samples, so we can see that perhaps what we should have had, or we used to have a couple hundred years ago. And the other point is what we do to the microbiota can actually impact our entire lineage, because you can only pass on what you've got. If it's not there, you can't pass it on. And if it's not a particularly healthy ecosystem, you may not want to pass it on. So I think it's one of the things that will filter into maternity care, is actually trying to make sure the microbiota is as healthy as possible, so that the bug will be inoculated with the best bacteria possible. And if you want to read more, The Good Gut by the Sonnenbergs, they're a research team from Stanford, I think. Great microbiota researchers, excellent book. And also Martin Blazer from New York Uni, researcher there, Missing Microbes. Both brilliant books. I think that's the end. But we've got time for questions, maybe. I don't know what time it is actually. Yeah. Any questions? Yeah.
Probably a bit of a multi-phase question, sorry, but
I like this, it's okay.
the first one would be like, how well understood are the actions of these bacteria on an individual level, rather than sort of a, oh well, they had 10% more of that and such things happen.
Yeah.
The second would be, are there any efforts or research underway in terms of, I guess, kind of getting an idea of what an ideal ratio might look like? And the third would be, is there anyone looking into actually being a bit more proactive and trying to modify or engineer existing bacteria to be more beneficial?
The last one's yes. The first question, I'll have to say again now, so I got lost in the other questions.
How well are the effects of them understood on an actual individual level?
Listen, some of these things we're very much learning. Like that Oxalobacter formigenes, listen, 15 years ago, I don't think we knew it existed. And so research is ongoing. Bugs like Faecalibacterium prosnitzi, 10 years ago, I'd never even heard of it, and it wasn't out there. And now we're realizing, it does this, it does this. When it's low, it's associated with, Crohn's disease patients have got low amounts, and Crohn's disease patients who respond to the medications and get better have got higher amounts than those that are non-responsive. So we're teasing those things out.
Oh, sorry, just like, I almost meant, when I said individual, I mean like almost on a chemical or a really low level as to why they do such things, rather than simply the higher levels correlate.
Yes, yeah, they certainly are. Yeah, listen, that's a bit beyond my level of interest, looking at the very specific nitty-gritty, but the research is certainly there. And you could look at Faecalibacterium prosnitzi, and they'll look at the cytokines and how it interacts with the gut immune system in myriad detail. The research is there, it's ongoing. It's just not in here. Yeah. Is there a second part?
Ratios.
Oh, yeah, so you mentioned that different cultures have different ratios, and that certain people even within one society do better or worse at certain things. Do people have any sort of idea of what might be a better ratio or a worse ratio or an ideal one?
No, people are still trying to tease that bit out. And with the new technology, as I said, there's new species that we didn't know existed before, and we're able to see what sort of genes they've got, what they're capable of doing. That's helping that. I think the current idea now is that the key thing is diversity and having good levels of some of those key beneficial species. Some of the other things we're not quite sure, but things like Faecalibacterium prosnitzi, another bug, Akkermansia muciniphila, when we've got those two bugs, it seems to correlate with a high degree of diversity and a lower risk of a range of Western diseases. Yeah.
I also have 3 questions.
Okay.
The first is, you didn't mention, I believe there's one theory around that contributes to this, is that we live in impoverished environments. We don't spend so long sitting outside with mud and things, and as kids we tend to keep them out of the garden, which we shouldn't. So that's
Yeah, there's two sort of, I suppose, microbial hypotheses. One is the disappearing microbiota hypothesis that Blazer has put out, which is, we've just lost this diversity. And the second one is the hygiene hypothesis, which is around that exposure to different things. And I think that's obviously important for developing normal immune tolerance and even perhaps introducing different species that at least interact with us for a period of time. Maybe some actually permanently stay there.
You didn't mention alcohol. I wonder if there was any impact on that.
Yeah, I should have actually. Not so good. The challenge with alcohol is that most of the research is done on alcoholics. What we don't know is what impact 1 glass of red wine or half a glass of wine at dinner 5 nights a week is. No idea, and probably minimal. But we know that people who have alcohol problems, who drink lots, it actually causes a massive reduction in diversity and a massive reduction in beneficial bug counts and an increase in gram-negative bacteria. And we also know that the liver damage that's caused in alcohol is actually caused by endotoxins, which is in the cell wall of gram-negative bacteria. So it's essential, and people argue that, and this is why you can take lactulose as a prebiotic or different probiotics that actually minimize the damage to the liver caused by alcohol, because it's changing the gut ecosystem. So yes, large amounts not good, small amounts not sure.
And I'm sorry, the third one is, as a stem cell scientist, we can make really nice gut structures now. Is anyone looking at colonising gut structures and to try and understand the interaction between the microbiota and the actual lining of the gut, because we can get crypts and
interesting. I'm sure there is, but it's a little bit outside my area.
A lot of this might be asking it in 5 years sort of stuff.
Yeah.
Well, as a stem cell scientist, it seems pretty obvious you could do some fun stuff with that.
Yeah, I reckon.
I was just going to ask about yeast. You haven't mentioned yeast at all, whether that has any influence on...
Like the ingestion of yeast? I think that would be minimal, because certainly like Saccharomyces cerevisiae, sort of baker's yeast, generally doesn't stick around for any great length of time in the gut, so it would be a transient sort of visitor. And the fungal microbiota is a new frontier that I'd say is just starting to be looked at. They've been really focusing on the bacterial components, and they're just starting to look at the viral components, and they're just starting to look at the fungal components. But it's turning out that there are actually a lot more fungi in there than once we once thought, too, and viruses too, for that matter.
You showed how long patients would have sort of quite severe antibiotics or combinations. What about something like, simple erythromycin or something like that? How long does the standard recovery from that?
Erythromycin, I can't remember off the top of my head, and I'm trying to turn it all into a database so you can just search antibiotic and it'll tell you what the researchers said in my spare time, which I don't have much at the moment. It's less, but I think I'd still follow that same procedure of taking the right probiotic alongside for 6 weeks afterwards, a prebiotic for 6 weeks, and then focus on those microbiota nourishing foods, regardless of what it is. Because even we can look at what that study showed, it's still not going to necessarily reflect how it's going to react to you, and that depends on how many different courses of antibiotics you maybe have had, or whether you've taken PPIs or NSAIDs at different times, or your alcohol intake, your diet. All those things play a role in determining the overall effect on you. So you're better off assuming that it's causing greater damage and treating it as such, I'd say.
How does this relate to mental health?
Well, you're right. In fact, they're starting to tease that out, and it's only been probably the last 3 or 4 years that they've started making the links between gut bacteria and mental health, and making links between anxiety and depression and gut bacteria, and using interventions like probiotics and prebiotics that shift the gut, actually improving depression scores and anxiety scores. So I think it's an area that's just starting to boom, and I think it's going to be obesity and mental conditions that'll be, and in connection with the gut for both of those, that'll be the next 5 years. There's going to be a tremendous amount of research done in those areas, because they're both widespread issues in Western nations.
Yeah, when you were talking about the reduction in types of bacteria, it made me think of the increase in depression and anxiety, and when you said that can be heritable to a degree, I was wondering if we're siphoning to that type of bacteria in a way.
It is a very interesting thought, because we are essentially, the mums are passing on what they've got. And if they've got a microbiota that's prone to depression, that's what they're passing on. And we know that with animal models, we can take that depression, a person's poo who's depressed or who's anxious, and introduce that into animals, and they become depressed or they become anxious. So there's certainly research that supports that idea. And just what we're not sure the best way of turning that around. And at the moment, I think following an approach like that improves diversity and improves levels of bugs that we'd see as tend to be sparked with healthier people would be the best approach. Yeah.
This is a really long shot, but I thought, what the hell?
Yeah.
Another two, I guess. First is, so you've talked about how diets changed in our society over time. That's definitely true. Another thing that's sort of changed over time is the disappearance of famine and other things like that. Are you aware if malnutrition or anything like that has an impact on this kind of stuff? Second question related is this whole same thing of diets changing over time and our lifestyle changing over time, and the elimination of famine is, there have been, I believe, epigenetic changes that people have talked about as possible reasons why things might be different now in terms of obesity and other things. things compared to the past. So I guess, do famines and malnutrition affect it, and is there any link between epigenetics and this at all?
The second one, yes, but it's not my area of expertise either. And the first one, I'm sure there is some, it would make sense. Yeah, listen, if you're on a very, very restricted diet, then that's going to impact what actually gets down there to your gut. And if it's mostly plant food, whole plant food in small amounts, some of that will still actually get down there. And perhaps their microbiota, certainly when they've looked at, I've certainly seen research on certain populations in Africa, and they actually had really quite healthy gut microbiotas. But I don't necessarily think it was people who were in a famine state, but there were certainly people who were probably more on that poverty line, but they were still actually getting all the plant. Essentially the diet was very high in fiber, pretty wide in diversity in that environment, and the ecosystem seemed to be quite healthy and diverse. But I think, yeah, if you put that to another step over, it would change.
It's really hard to comment, it's really hard to navigate. Like if you go down a health food aisle in a health food store and you see 20 probiotics and you know they range from $20 to $100 a bottle, and you're so confused, and then there's kombucha in the fridge, and you can make your own kefir yogurt, and it's so hard to understand what you should be doing and what you should be eating instead, or what can you take as a tablet to compensate for your crappy diet. I find it really overwhelming.
Yeah, it's tricky, and it's tricky for health professionals too, for that matter, the same sort of issue. And I mean, it's when I actually developed a tool called the Probiotic Advisor for that reason, for health professionals, so they can actually look at what the shelf says and, hey, what does that actually have in it? What's the research on that particular strain in that supplement, so they can actually follow that through.
Can we use that, or is that just a
it's open. It's a subscription database, so I can recoup some of my costs I put into it. But it's there for essentially for people can actually, look at. They read a study saying this particular strain is useful. Where can I find that? It tells them that. If you can type in a condition like antibiotics, and it tells you what strains have got the best research evidence, which ones have meta-analysis data, which ones are just randomized controlled trials, which ones are open label trials, and it has a brief explanation of the study to try to make it easier to navigate through that, because it is a maze. And listen, for me, I've been 15 years researching this area, so I can navigate it pretty well, but it's took me a long time before I was able to navigate it well. So I'm trying to make it easier.
So just on that, I sort of got the impression that you were saying supplementation with probiotic, unless you had a course of antibiotics, is useless. Is that?
I probably didn't want to give that exact impression. That different probiotic strains have got different actions, and I think this is some speed up gut transit time, some slow it down, some have a healing effect on the gut mucosa, some actually help regenerate a small intestinal architecture after damage. I use probiotics as tools as a clinician all the time, but I'm using the right one that's got the action that I'm after, just like you use ACE inhibitor if you want to decrease blood pressure. And there are some probiotics that decrease blood pressure actually by producing an ACE inhibitor compound in the gut. So you choose the right strain that actually has the action that you're after. So I use them widely, but I think that if you're after trying to create the healthiest gut ecosystem possible, I think probiotics actually have a minor role there. Besides during insults, if someone has to be on PPIs long term, sometimes they do, or non-steroidal anti-inflammatories long term, or chemotherapy, radiotherapy, listen, I think they're extremely useful to actually help decrease the side effects, decrease the change in diversity, decrease antibiotic resistance. But if you really want to make your gut healthy, do it with food, with prebiotic supplement. That's what the research was saying.
Just talking about the clinical application of these things, a straw poll of my colorectal surgeons would say that they knew nothing about nutrition at all.
Yeah.
My doctor pumps me about it. How do we get sort of traction that you need to be able to do this? Do you have any ideas about that? I mean, I think colorectal surgeons should probably have some idea about it. I want to.
Yeah, I agree. I'm not sure, because the research is being published in gastroenterology journals. It's not like it's in far hard to find stuff. But I think if it's not part of undergraduate training, we know that it means that it doesn't really get looked at post-grad. And whether there's some way of trying to make sure that it's part of their post-graduate education, I'm not sure, because they are really quite behind. Particularly in Australia, like when I went to Italy, it's like all these gastroenterologists all talking about dysbiosis and how we can do this dietary thing.
I was really struck by that.
Like, well, it struck me too, because here I'm at this conference and there's these guys working at the local hospital, and this is what they're actually recommending to the patients is diets like that, prebiotics and probiotics, and it was just like, wow, Australia is really behind. It is in this area. And it means, and clinicians need to get that information. Like even GPs need that information, because it's not as if there's not evidence there anymore. You could say that 30 years ago that there wasn't the evidence there, but sometimes now we've got meta-analysis level data. It's good data. That should be changing our practice because it means that our patients get better outcomes and less side effects.
My GP is really interested in it, but just doesn't know if it makes a difference. It's really frustrating.
Yeah, and it does.
I know it does.
But
I know it does.
But you know, but they're busy too, and their lives are so full. It's hard for them to look beyond. And I'm just saying that as a bit of an excuse, but it's, in some ways it's not, because
and it's so much easier to give a pill than to give dietary information, because people find dietary information really confusing.
yeah, and it takes time.
It takes
Like
time.
for my patients, I spend an hour and a half that first consultation, taking case and doing dietary stuff. You know, and most practitioners in the mainstream medicine do not have that. Will not give that amount of time per patient, because you have to explain stuff. You have to answer questions. You have to make sure they got it. So.
Okay.
Thanks so
All right,
much.
thanks.
Thank you.